हेलो एवरीवन, वेलकम बैक टू अनदर वीडियो फ्रॉम shomus biology। और इस वीडियो में अलग से हम बात करेंगे मैथ प्रॉब्लम्स फ्रॉम जेनेटिक्स। सो बायोमैथ प्रॉब्लम्स फ्रॉम जेनेटिक्स, एंड अमंग द बायोमैथ प्रॉब्लम्स फ्रॉम जेनेटिक्स, particularly from genetics probability, there are different kinds of math problems from genetics. Genetics chapter is mostly conceptual, and in CSIR NET, GATE, ICMR, DBT, they ask a lot of math questions from genetics. So we'll prepare them, particularly the genetics probability problem in this particular lecture. English हिंदी दोनों लैंग्वेज में ही होगा। इस सीरीज की हर एक वीडियो दोनों लैंग्वेज में ही हो रहा है मिक्स करके। So I'll explain both. Okay, so we have this first question, and this is a typical representation of a type of question which is generally asked to make you confused, and because a lot of information is given in this question, and in reality only one or two informations are actually required. So what is given here is that I'll read it from a little distance, otherwise I cannot see. In mice, coat color is governed by two genes, A and B. Gene A, when dominate, it dominates and suppresses the expression of gene B. Thus, a mouse appears albino whenever it has at least one dominant allele A. So actually the color of the mouse is governed by B gene, but whether the color will be added or not is regulated by A gene. B gene क्या करता है? कलर ऐड करता है। तो B कैपिटल B है तो ब्लैक कलर होगा, स्मॉल B है तो ब्राउन कलर होगा, ऐसा ही। इसी कैपिटल B रिजल्ट्स इन ब्लैक, और स्मॉल बी अगर होता है तो दैट विल बी रिजल्टिंग इन ब्राउन. So that is very simple. So let me explain this question. It stated that capital B, it will be the color black, and if this is small b, then it is the color brown. Okay, this is done by this B gene. Okay, capital B, small b also cause black because that is the heterozygous nature, but capital A dominates over B, dominates over capital B. So capital A is a epistatic gene. Capital A is an epistatic gene over the B gene. B gene is a hypostatic gene. So B gene is being regulated by A gene. तो यहाँ पे कैपिटल ए जीन है जो एपिस्टेटिक जीन है, वो कैपिटल बी जीन को इनिबिट करता है। मतलब कैपिटल ए जब तक मौजूद रहेगा तब तक कैपिटल बी कोई भी कलर ऐड नहीं कर पाएगा, क्या वो ब्लैक हो चाहे वो ब्राउन हो। अगर ए है तो ब्लैक ब्राउन कोई भी कलर ऐड नहीं होगा। जब कैपिटल ए एब्सेंट है, व्हेन द कैपिटल ए इज एब्सेंट, देन ओनली यहाँ पे कलर होगा। मतलब द ओनली टाइम कलर विल बी विजिबल इन माउस इज द जेनोटाइप स्मॉल ए। जब जेनोटाइप स्मॉल ए होगा तब जाके कोई भी ऐसा कलर ब्लैक या ब्राउन ऐड होगा, बट जब कैपिटल ए रह जाएगा तो कोई कलर ऐड नहीं होगा, नो कलर। और कोई भी कलर ऐड नहीं होगा उससे माउस जो है उसको हम बोलते हैं कंडीशन, जिसको एल्बाइनो कहा जाता है। तो एल्बाइनो कंडीशन में वो रह जाएगा। एल्बाइनो मतलब कंप्लीटली व्हाइट, कोई भी कलर नहीं आता। ठीक है, तो दैट इज द एपिस्टेटिक इफेक्ट। नाउ यू अंडरस्टूड हाउ दिस एपिस्टेसिस, हाउ दिस प्रोसेस वर्क्स. Now we can take this understanding a little deep in order to explain it in the epistatic manner. I'll also do that, although it is not necessary to answer the question, but I'll do that because I want you to understand the mechanism. So there are four situations. One situation is something like this. There are two loci, one is for the A, another one is for the B, and the presence of the genes is something like that. 9 individuals like this, 3 individuals there will have something like this, 3 individuals will be there with have something like this, and 1 individual will be there with the genotype this. Right, now remember, among this 4 segment 9:3:1 ratio, that is the phenotypic ratio of Mendel, and what we know here is that if capital A and capital B both are present, what color will be visible? Try to understand. If capital A is there, no color, so that is albino for you. Again this one, capital A is present, no capital B, so again albino. Okay, for sure. Third one, capital A is absent, capital A nahi hai, so when there is no capital A, then color will be added. Now again, based on which gene is present, which like whether the dominant allele or recessive allele of the B is present. Dominant allele means this is black color, and this is recessive allele. Capital A is absent, capital A nahi hai, तो small b की मतलब हुआ brown color. So in this particular example, how many albino will be visible? 12 albino. 9 plus 3, 12 albino, 3 black and 1 brown will be visible. This is a typical example of an epistasis event, right? Now, now let us talk about the question. The question stated, if two heterozygous black mice are crossed, what is the probability of them producing a brown offspring? If two heterozygous black mice are crossed, what is the probability of them producing brown offspring? मतलब parents की बात करें तो वहां पे दो black mice आएगा, दोनों का ही जो genotype होगा वो heterozygous होगा, that is capital S small a capital B small b. अगर वैसे क्रॉस होता है, मेटिंग होता है, तो प्रोबेबिलिटी क्या है कि वो एक ब्राउन ऑफस्प्रिंग को जन्म दे? व्हाट इज द प्रोबेबिलिटी दैट दे विल गिव राइज टू अ ब्राउन ऑफस्प्रिंग? ओके, for that we'll do the calculation. See, now the condition is capital A small a capital B small b. We cross it with capital A small a capital B small b. Now when this kind of crosses are present, I know many of you get puzzled like what to do, what not to do. Now the easiest way to solve these problems is something like this. There are two genes A and B, so separate them. So A को separate करो तो A के साथ A के क्रॉस करके देखो कि क्या रिजल्ट आता है between the cross. We get one option, capital A capital A, capital A small a, capital A small a, small a small a. That is the option that we get. If we cross between the A, अगर A के बीच सिर्फ A जीन के बीच हम क्रॉस करते तो ये रिजल्ट आता. अब B के बीच अगर क्रॉस करें तो क्या रिजल्ट आएगा वो भी देख लेते हैं. कैपिटल B स्मॉल b, वही चीज होगा कैपिटल B स्मॉल b. तो एक कैपिटल B, ये एक कैपिटल B स्मॉल b, ये भी कैपिटल B स्मॉल b और ये होगा स्मॉल b, राइट? तो सेम कंडीशन है, दोनों में ही सेम है. नाउ फ्रॉम दिस विल डू द कॉम्बिनेशन, राइट? सो व्हाट इज द प्रोबेबिलिटी ऑफ दिस इंडिविजुअल वैल्यूज? द प्रोबेबिलिटी को कैलकुलेट करते हैं. देखो, सो वी वांट ब्राउन ऑफस्प्रिंग. नाउ टू बिकम ब्राउन ऑफस्प्रिंग, व्हाट यू मीन बाय टू बिकम ब्राउन? ब्राउन ऑफस्प्रिंग का मतलब ब्राउन ऑफस्प्रिंग कब होता है? जब कैपिटल ए एब्सेंट होता है, देयर इज नो कैपिटल ए, देन ओनली ब्राउन ऑफस्प्रिंग विल बी देयर, राइट? Right? The only time when there is a brown offspring when there is small a. तो हमें पता करना है कि small a जीनोटाइप का probability क्या है. What is the probability of getting small a? And here we can easily see that 1 out of 4 is only small a. So for brown we need small a and the probability of is that is 1 out of 4. Clear? First part is clear. Next, to become a brown you also need what? कैपिटल B रहेगा तो ब्लैक हो जाएगा, स्मॉल b रहेगा तब ब्राउन होगा. तो टू गेट ब्राउन we need small b as well. So what is the probability to get small b genotype? In this cross again the probability is 1 out of 4 to get a small b. So small b is probability to get 1 out of 4. And we knew it that from this brown offspring is only possible if there is no capital A, no capital B in either of the locus. कैपिटल ए भी नहीं चाहिए, कैपिटल बी भी नहीं चाहिए, तभी जाके ब्राउन ऑफस्प्रिंग जन्म लेता है. अगर ए, कैपिटल ए या फिर कैपिटल बी में से कोई भी मौजूद हो तो ब्राउन ऑफस्प्रिंग कभी नहीं होगा. तो व्हाट इज द प्रोबेबिलिटी टू गेट स्मॉल ए 1 आउट ऑफ 4. व्हाट इज द प्रोबेबिलिटी टू गेट स्मॉल बी 1 आउट ऑफ 4. तो टोटल प्रोबेबिलिटी क्या होगा? हमने तो सेपरेट किया था ना, ए और बी का अलग करके देखा. सो टू गेट अ टोटल प्रोबेबिलिटी we need to apply the rule of multiplication. We need to multiply, right? कौन सा चीज? प्रोबेबिलिटी ऑफ ए और प्रोबेबिलिटी ऑफ बी को मल्टीप्लाई करेंगे, जो कि 1 आउट ऑफ 4 इंटू 1 आउट ऑफ 4, 1 आउट ऑफ 16 होता है आपका आंसर. सो द आंसर इज 1 out of 16 because we need to multiply both the probabilities to get a small a, that is 1 out of 4, to get a probability of small b, that is another 1 out of 4, and we get the answer 1 out of 16. So here the option B stating 1 out of 16 is the correct answer for this question, where we have the epistasis in mice. How exactly the values will be depicted? This is how it is depicted. You can take a screenshot and you can rewatch it couple of times if it's not clear in one go, but this is how the whole process works. In epistasis problems, it's very important that you'll be able to do this. You'll be able to understand the genotype and the number of individuals will have the genotype for 9:3:1 ratio. Only when you understand it, you will be able to answer all the mathematical question from this topic. Okay. Let's move on to the next question now. The next question here we have, and that is from the sex-linked inheritance. Okay, sex-linked inheritance. Hemophilia is a sex-linked recessive trait. A non-hemophilic woman whose father had hemophilia marries a non-hemophilic man. What is the probability that they have a son with hemophilia? Okay, so again another probability question. Now, if in this question it is told that hemophilia is a sex-linked recessive trait, they may or may not tell you that. But you need to know that hemophilia, color blindness, these are all X-linked character. So it is transmitted, it's not transmitted, we say it is transferred from mother to son more often and from father to daughter, okay, because X is the chromosome that carries the gene for hemophilia and color blindness. So now a non-hemophilic woman, so you see that this is a woman who is non-hemophilic. Woman means XX and men means XY. So non-hemophilic men, okay, whose father was hemophilic. So H is for hemophilia, so father was hemophilic, marries a non-hemophilic man. So this is a non-hemophilic man, this is also a non-hemophilic woman, but the father of this woman was hemophilic. So we know that from father, here is the father, father gives X to daughter, right? Father gives X to daughter and father gives its Y to its son. So if this mother, this is a normal individual, right? No hemophilia to this woman, but one of the X this woman got from her father, and her father must have given one XH. The other X she must get from her mother. So that is a normal X. So this individual, although seemed normal, but in reality is not that normal, is actually a carrier of hemophilia disease. So one XH and another X. तो ये जो महिला है एक XH फादर से ड्रॉ किया है, क्योंकि फादर एक X ही अपने डॉटर को देगा और Y जो है वो अपने बेटे को देगा, सन को देगा. तो यहाँ पे ये जो मदर है इनके पास एक X जो H है वो फादर से आया है, तो इसका मतलब है मदर जो है नॉर्मल तो है बट शी इज अ कैरियर. And the other individual, the male here in this case, is completely normal. There is no doubt about it, a normal male. Okay? So now if that is the cross, then what is the probability that they have a son with hemophilia? So now you need to simply calculate this idea. So we have XH, X, then we have XHY, then we have XX, and we have XY. 4, we drag it and it's going to look like this: XHX, XHY, XX and XY. So here you can see clearly कि ये जो female, this is a female, this is a carrier female, but in reality carrier are also normal, not the disease carrying one. This male is infected, this is again normal, this is again normal female. So this is also normal. So what is the ratio of abnormal to normal? Only one infected out of 4. This one, and this is a boy, this is a son. So what is the probability that the son to have hemophilia? The answer is 1 out of 4. 1 out of 4 in this case, you can see. So here you can clearly see that among men, the question is what is the probability that the son have hemophilia? So you can see the son, infected one is 1 out of 4, but there are 2 sons. This is one son, this is another son. So between 2 boys, how many of them are hemophilic? Between 2 boys only 1. Between these 2, only this boy got the hemophilia, because the other boy gets the healthy copy of X from the father, the other healthy copy from the mother. So the healthy copy of X from the mother and the Y from the father, obviously. So they both, between these 2 boys, what is the percentage showing hemophilia? Only half of it. 2 boys, one of them has hemophilia, another one without hemophilia. So probability is half. So the option A should be the correct answer in this case. Half of the boys have the probability, half of the boys have the probability to have hemophilia in this occasion. Clear? Alright, so now we are moving on to the third question here, and the third question is a coat color in dogs. Coat color in dogs. In dogs, the coat color is influenced by two genes, capital H and I. The dominant allele capital H produces a black coat, but if at least one I allele is present, it suppresses the expression of H, resulting in a golden coat color. So again we are looking at gene interaction. If a black dog of a genotype HHII, capital H, capital H, capital I, small I, is crossed with a golden dog of small h, capital I, small I, what is the probability that they will produce a golden offspring? So again a probability calculation, but here also we are seeing that there are gene interaction. यहाँ पे भी gene interaction देखा जा रहा है, and the gene interaction is like between the two genes. One is capital H, another one is I. Now the question is, the capital H, the dominant form, capital H form, produces black coat. Okay, it normally produces black coat, but when capital I is present, it inhibits the capital H's activity, and if capital I is present, it causes, even if one I allele is present, one capital I is present, it is repressing the black coat color and it allows the coat to be golden. So the thing is, if capital I is present, it marks golden. Capital H never marks black alone. It marks black only when there is capital I is not present, when there is only I is present as small I, small I. So again we need to rearrange this data set. हमको फिर से इस data set को arrange करना है epistasis के हिसाब से. मैंने पहले भी एक एग्जाम्पल हमने देखा है जहाँ पे एपिस्टेसिस की कैलकुलेशन के हिसाब से करना था, यहाँ पे भी हमें वही चीज देखना है. और यहाँ पे भी देखें कि कैपिटल H और कैपिटल I 9 इंडिविजुअल्स आएंगे, फिर 3 आएंगे कहाँ पर, कैपिटल H स्मॉल आई, फिर 3 इंडिविजुअल्स आएंगे यहाँ पे स्मॉल h कैपिटल I, और कुछ भी हो सकता है, और एक इंडिविजुअल होंगे स्मॉल h स्मॉल आई. So this is again the distribution as per our understanding of epistasis or gene interaction. Now from this gene interaction what we need to know is basically this, where capital H and capital I both are present, then what will be the color added? If I is there, it causes golden, so golden. So this is I is there, so it will cause the formation of golden, right? And if there is no capital I, then only capital H shows his impact, that is the color black. And here capital I is not present, so obviously the H can impart the color, but here there is no capital H, so again it will be golden. It won't be black again, it will be golden again. Okay, so that is the idea. So this is the simple idea of the epistatic event and the interaction between capital H and capital I. इस हिसाब से H and I की भी interaction हो रहा है. Now we need to see the genotype. दो genotype दिया गया है यहाँ पे, capital H, capital I, small I, capital I, small I, and what will be the probability that they will produce a golden offspring. तो इस दोनों के बीच देखो capital H, capital I, small I cross हो रहा है with small h, capital I, small I. So this is the cross that we are looking at, right? And in this cross what we are trying to figure out is that what we are getting from this cross is that here all of them will have capital H, small h. So between H, अगर H को एक साथ लेते हैं तो उस बीच एक ही genotype possible है होना, that is capital H, small h. And between capital I and small I, what are the possibilities that we might get? We might get capital I, we might get capital I, or small I. We get this three separate probabilities from this aspect, three separate probabilities. Okay, now three separate probabilities we get here. So there is a chance यहाँ पे जैसे कि एक chance क्या है कि हर every single individual will be capital H, small h. So heterozygous for that. Now the game changer would be the presence of capital I or not, if capital I is present or not. Okay, so if capital I is present in all these cases, you can see that the capital I is present. So if capital I is present here, that will lead to the golden offspring. Capital I is also present here, lead to the golden offspring. And here small I is, so there is no capital I in only one occasion. एक ही जगह है जहाँ पे small I तैयार हो रहा है, एक ही genotype यहाँ पे बन सकता है जहाँ पे स्मॉल आई है, मगर उसके साथ है कैपिटल H स्मॉल h, ये है कैपिटल H स्मॉल h। अब बात ये होता है कि कैपिटल H प्रोड्यूसेस बैक कोट कलर, but if at least one eye is present it will suppress, but this is only one occasion when there is no suppression done, because capital I is absent here, capital H is present, capital I is absent. As capital I is absent, so if there is no capital I, capital H is present, that occasion the color would be black. So that color will be black only. So apart from that, rest of the color will be golden, right? So what is the probability for that? The probability for that you need to count, because the probability of capital H small h is always 100%, but probability of II and II, यहाँ पे golden होने की probability थी, how much? To get a golden, the probability of having a golden individual here is again between these two, 3 out of 4. So 3 out of 4 is the probability to become a golden. So the total probability of having a golden offspring here would be 3 out of 4, that is option B, right? So 75%, 3 out of 4 means 75%, because you see that the probability between I and II, that's what we are calculating, right? That's what we are calculating, and we can calculate it fairly enough, that capital I, capital I, capital I, small I, capital I, small I, small I, small I, right? So 3 of them, 3 out of 4 gives us the capital I, at least one capital I, that means golden, and one is the case where there is no golden. But on the other side we have capital H small h, and capital H small h can influence the color of black, right? So that's why this one, only this individual will give a rise to black color. Remember, this genotype, individual with this genotype, capital H small I will give a rise to black, rest will not give a rise to any black color. So as per this cross, it will be 70% of the population showing the golden color, right? Rest all of them showing the golden color. Okay, here the ratio comes 13:3 based on the double cross that we know here, the in case of Mendelian ratio, which is altered, epistatic ratio, but this is something that we get as a result. It might feel little complicated at first, but again rewatch this part couple of times in order to get a clear idea, and you'll be able to answer this with ease. Now the last question here, and in this question again another probability problem. If plant with genotype capital S small a, capital B small b self-pollinated with self-pollinated, where capital A and capital B are not linked, then the probability of getting A, B, B genotype will be all capital. So this is very easy question, you know, before, like, this question should be provided earlier, because we have discussed about complicated problems earlier, and now we are into a smaller and simpler problem. This simpler problem that we have is very straightforward. Like, we have this capital A, capital B, small b, okay, self-pollinated means we have same genotype to be accustomed to it. Now when we have this kind of crossing, the easy way to find out the next probabilities is to divide the number of genes. बहुत आसान हो जाता है जब अगर आप डिवाइड कर लो, दो जीन्स है A and B, इसको डिवाइड कर लेते हैं, अलग अलग से हम पनेट स्क्वायर को देखते हैं. देखो वैसे तो एक साथ भी देख सकते हैं, मगर अलग अलग से देखने से आसान हो जाता है. कैसे आसान होता है? देखो कैपिटल A और स्मॉल a, इसके साथ है कैपिटल A और स्मॉल a, ठीक है, और इस बीच अगर देखते हैं 4 ऑप्शंस आएंगे, ठीक है, एक तो कैपिटल A आएगा, एक ये कैपिटल A स्मॉल a आएगा, एक ये कैपिटल A स्मॉल a आएगा, और ये स्मॉल a आएगा. These are the 4 options. For B also the same situation, right? Capital B, this will be capital B small b, this will be capital B small b, and the end one will be small b, right? So what we are getting from here, what we are getting is clear results. So now what we want this genotype, individual, number of individuals with capital A, capital B genotype. So from this whole population, what we want to figure out? Capital A. What is the probability of capital A? From this B side, we want what is the probability of capital B, right? So what is the probability of capital A? कितने बार आया capital A? एक बार out of 4. So 1 out of 4 is the probability of getting a capital A. Now व्हाट इज द प्रोबेबिलिटी ऑफ गेटिंग कैपिटल बी? कितने बार कैपिटल बी आया है? टोटल में से 4 बार टोटल है, इसमें से 1 बार आया है, तो 1 out of 4 is the probability of getting a capital B. Now the total question asked for the probability of this genotype, capital A, capital B. So what will be the genotype, what will be the probability to get this? The probability to get this वही है, मल्टीप्लाई करना है आपको. 1 out of 4 is capital A, 1 out of 4 is capital B. And if you do the multiplication, you get the overall probability. This is known as rule of multiplication. And we use rule a lot. We use this multiplication rule a lot in genotype problem. यहाँ पे 1 out of 16, यहाँ पे लिख देते हैं option C. नहीं था, but I'll put it here. The option C, 1 out of 16. That is the correct answer. 1 out of 16. That is option number C, where we know that the probability of getting capital B is lesser 1 out of 16. 16 में से एक बार ऐसे सिचुएशन होगा, एक जीनोटाइप आएगा जो कैपिटल B तैयार करेगा. So for any kind of problem, if they ask you a question that instead of having capital B, they told that what will be the probability of getting this capital A, small a, capital B. तब क्या होता, right? Think about it. तब क्या होता? So first of all, capital A, small a की probability क्या है? देखो 2 बार आया out of 4, so 2 out of 4, or I can say half, right? And for capital B, we know it's 1 out of 4. So the probability to get capital A, small a, capital B would be half into 1 out of 4. That will be 1 out of 8, right? So let's say something else. What is the probability of getting this? Capital small a, small b. उसका क्या probability होना है, right? What is the probability? Second, to get a small a, what is the probability? कितने बार आया? 1 out of 4. Small a, small b also 1 out of 4. So again the probability would be 1 out of 4 into 1 out of 4, 1 out of 16, 1 by 16. So this is how you solve probability problems for gametes and genotypes in Mendelian and non-Mendelian inheritance. There is no big deal about it. It's very simple to solve. The math that is required is only multiplication, addition, and in some cases it's just square, calculation of square, right? That's what you need to understand. We will solve some more problems later on in a separate lecture where we talk about the rule of addition and square also. But for now, multiplication works for the most of the problems from these probability questions in CSIR NET, GATE, ICMR, DBT, GRF exams and IIT JAM. So genetics is a chapter where 80% of the question will be this mathematical or analytical, right? So for genetics, you need to have this book, Biomath Handbook. And actually, if you look at this whole book, it's approximately 250 to 60 pages book. And with this near about 260 pages book, most of the book is consisting of biochemistry math problems and genetics math problems and cladogram problems, because these are the three problems area, these are the areas from where CSIR NET ask a lot of math problems. So if you get inside, the genetics chapter starts at 67th page and ends in 128th page in edition 2. You might get edition 3 books. In that case, the page number might vary a little bit, but we increase the number of question in every single edition. So genetics की problem की बात करें तो genetics book 67, page number 67. Here you have the genetics math problems, and from here you will get Mendelian, non-Mendelian problems, genetics gene mapping problems, pedigree analysis, probability, risk calculation, additional problems, HFR mapping, bacterial genetics problem, co-transduction frequency problems. So you'll get all these problems. Here are all the normal Mendelian genetics and inheritance problems, आप देख सकते हो, right? And if you want to practice more from the, let's say, if you want, so these are all non-Mendelian inheritance problems. Answer with explanations are also provided. It's not just answer, but explanations are also provided. And we have practice problems also. You will get the gene mapping problem. These are the gene mapping problems starting at page number 94 here. So these are all gene mapping problem. You can see that sample problem with answers are provided. These are the gene mapping problems. You can see in this side gene mapping problems are going on, right? Gene mapping problems. So all these gene mapping problems are there. And not only the gene mapping problems are there, but also their answer explanations. It's not likely there are one or two questions. There are more than 5 questions are there from every single topic that we discuss here, right? आप पेज scroll करते करते थक जाओगे, इतना problems आपको मिलेगा. This is another gene problem, gene mapping, it's continuous gene mapping. Now started with pedigree. यहाँ से pedigree start होता है. Pedigree problems, you can see pedigree, all these problems, pedigree problems, never ending pedigree problems, बहुत सारे problems आपको मिलेंगे, and you will get to answer them, you will get to know these problems. These are bacterial gene mapping problem, HFR mapping problem, these are also there, the HFR problems, right? So all these problems you will get from this book, that is Biomath Handbook. Okay, and there is a QR in every single chapter. If you scan them, it's going to open up the videos explaining this book. So this is a one of a kind book which you can get from us and us only, but it's not to be sold alone. You need to get the complete study package, that is of 3,900, including the delivery charges to your place.